Hello everyone, this is Katish here. In this video, I'm going to go through the AQA Biology Paper 1 Higher Tier from 2025. Question number 3 is on the topic of cell biology. A student investigated osmosis in pieces of potato. This is actually the required practical on osmosis. This is the method used. Cut a piece of potato into a cube. Record the mass of the cube. That will be the starting mass. Place the cube into a beaker containing 0.4 mole per dm cube salt solution. After some time, remove the cube and record the mass. So that's the end mass. Calculate the change in mass of the cube. Repeat steps 1 to 5 using 2 more different concentrations of salt solution. Evaluate the student's method. For 6 marks, you should include how the student could improve the validity of the results. Now, how would you improve the validity of the results? You'd want to repeat it. You would want to take average values. You'd want to use different concentrations, not just 2 more concentrations. You'd want to use a control. So let's have a look at the student's method. Cut a piece of potato into a cube. It doesn't say is it coming from one potato or from different potatoes. And it's only talking about a cube, not a lot of many cubes. It doesn't give you the size, which doesn't really matter as long as it's the same size cubes. Record the mass of the cube. It doesn't say, again, whether to peel the potato or not as well, because that wouldn't matter. Place the cube into a beaker containing 0.4 molar solution. It doesn't say how much volume as well. After some time, they're not actually mentioning the time as well. How long for? Is it 5 minutes or 10 minutes? Remove the cube and record the mass. Now, before you record the mass, you would want to blot dry the cube so that there's no excess water on top of the cube, because that would add to the end mass. Calculate the change in mass of the cube. Now, calculating the change in mass of the cube is not enough. You need to work out the percentage change in mass, because the starting mass might be different. It's not always going to be the same mass for all the cubes. Repeat steps 1 to 5. So they're not mentioning about repeating the same concentration at least 2 more times to get an average as well. And they haven't actually talked about mentioned the different concentrations that they're wanting to test. And 2 more concentrations is not enough. At least 4 more concentrations would be good. They haven't mentioned about comparing it to adding the piece of potato into pure water or distilled water, so that you can actually compare it as well. So here's the answer. Now this answer is actually quite long. Now looking at the marking scheme, they are asking, this is what the marking scheme is actually expecting students to get 5 to 6 marks. You need to have a strong judgment, strongly linked and logically supported by a significant range of correct reasons is given. So when they say significant reasons, they are looking for a lot. So it doesn't mean that you have to write all of this. The strengths are the same potato was used. We are assuming it says cut a piece of potato. All the potato pieces were cubes. The mass was recorded before and after. Change in mass was calculated. Different concentrations of salt solution were used. They did mention at least 3 concentrations. The improvements: use the same potato for all pieces, because the student may not have used different potatoes, Remove the skin or peel the potato, because the skin may still be present. Blot dry the cube before measuring the mass, because cube will have excess water on the surface. Use same cube size or surface area of potato piece each time. because it's not controlled or specified in the method. Repeat the whole investigation or at each concentration because there were no repeats to spot anomalies. Calculate the mean from repeats because there was no mean calculated. Immerse the cubes for the same time for each piece to be in the solution because length of time in the solution was not specified. Control the temperature. That's also important because osmosis can be affected by temperature. Using a water bath because temperature of solution is not controlled or specified. Use the same volume of solution for each piece of potato or ensure all pieces are completely submerged because volume of solution was not specified or controlled. Calculate the percentage change in mass because it was not calculated, or you could say because each potato piece could have a different starting mass. Include a control experiment with boiled potato because this was not done. Increase the number of concentration of salt solution used because only 3 different concentrations of salt solution were used, and you can actually specify the different concentration. They used 0.4. So you could start with 0.1, 0.2, 0.3, 0.4, 0.5 as well, and with distilled water as well. Why would a potato cube placed into distilled water increase in mass? Well, if it was placed in distilled water and it increased in mass, that's because water moved in. The solution in the potato cells was less concentrated than the distilled water. Distilled water is the lowest concentration, so that can't be it. The solution in the potato cells were more concentrated than the distilled water. That seems more plausible. The volume, and the next one, volume of water. The volume of water doesn't actually matter here, it's the concentration that matters. Active transport is another process used by potato cells. Give one wave. Active transport is different from osmosis. Active transport requires energy. You could also say it transports substances against or up a concentration gradient. And osmosis is the movement of water, whereas active transport involves transport of mineral ions. That brings us to the end of question number 3, and I will see you in question number